So this is a PhD student from the University of Florida's Plant Breeding Graduate Program. He is a National Science Foundation Graduate Research Fellow. His work in the strawberry breeding program focuses on the intersection of phenomics and machine learning. Specifically, he utilizes near-infrared and multispectral imaging to develop non-destructive methods for evaluating fruit quality. So to present his research on these complex parameters, please join me in welcoming Xavier Tacker.
Okay, I want to try something. Howdy. Okay, cool. I was getting the emails the last couple of weeks, and I was like, they keep saying howdy, but now I see it is a thing. Well, thank you guys so much for having me. Yeah, I'm from University of Florida. I'm a second-year student. I work in the strawberry breeding program there, and I'm finding out you guys don't do strawberries here, so I'm going to get to teach you a little bit about what we do and how we're using imaging and sensing to improve our fruit quality evaluation. Oh. There we go. Okay, cool. So we're going to break this down into a couple of major points. First, I'm going to talk about what I actually mean when I say going beyond the breeder's eye, and that's using the electromagnetic spectrum to actually get rapid non-destructive phenotyping and working towards selection with that. I'll break that down into a couple of major objectives, and then I'll get a little bit philosophical and Silicon Valley on you, and I'll talk about a couple of my ideas and things that I hope to do with AI as we get more data. So quickly, you'll find me next Saturday at the Florida Strawberry Festival. It's a big deal. I found out that our breeding program that's been around since 1948 is a very big deal in Florida. We grow about 16,000 acres, and 90% of the varieties are from UF. Another thing that's really exciting for us, I don't have to worry about G by E. Our actual area that we breed for is within about 40 miles of our location. So it's pretty nice, but you got to deal with the octoploid because everything's got gives and takes, right? And then if you do the math, we are a winter production crop. So yes, I should be at work right now. I will be at work when I get off the plane on Saturday. So let's talk about what you see and what you can select. Traditionally, we phenotype things by hand, especially for fruit quality, and that's going to be, your flavor chemistry, as well as shelf life, which I'll talk about in a little bit. This is relatively slow. It can be subjective, as we talked about the ratings a little bit earlier, and it's also predicated on human training. And unfortunately for us, the people that are most trained to do this kind of work also happen to be the breeder, the assistant breeder, the quantitative geneticist, and a postdoc. So a lot of people that have a lot of places to be at once, which means that our throughput on these traits is really, really slow. Well, thanks to advances in cameras and robotics, we can use drones, and they work really well in our program for traits like yield. But when you're flying at about 45 feet in the air, and the architecture of your plant is basically occluding all of the fruit tissue, you're trying to make predictions on fruit-related traits using vegetative-related tissue. Not going to work out too well. So we are splitting our traits that I'm going to be talking about today across the electromagnetic spectrum that you might remember from chemistry or physics class. In the visible spectrum, you have things like shape, size, color, gloss, a couple of different features like the seeds or the achenes, as well as the leaves. And then in the near-infrared spectrum, you have everything that has to do with sensory, and then I'll also use that for predicting gloss. To do this, we use a near-infrared spectrometer from Viavi. It's about this big, and we can take it into the field. The novelty of this, as I'll talk about in a bit, is that you can actually scan directly onto the fruit, and you don't have to rely on drones. It's pretty quick. It's about 6 times faster than phenotyping some of our traits. So let's talk about the actual game plan for this. This work that comprises my dissertation splits into 3 objectives, but I really think it's just 2, because 2 out of the 3 are using the NIRS, but to predict different things. Objective 1 is to model our actual breeder sensory evaluations, so learning from breeders and using it to approximate their selections. And then objective 2 is looking at the actual discrete latent chemistry factors that influence flavor. And then objective 3, using computer vision as well as the near-infrared spectrometer to actually try and automate a shelf life pipeline within our program. It's not something that we've done yet in the program. We've never really looked into shelf life, and it takes a lot of time. So I'm hoping to use computer vision and NIR to actually speed that up. So, it's no surprise. You have to learn from plant breeders to become a plant breeder if you are a NIRS device. So a typical workday is me out in the field. I get a fruit, I scan it with the device, I pass that fruit along to the well-trained, well-acclimated breeders. They're able to evaluate flavor on a 1 to 5 scale. And then I use partial least squares regression to actually predict their evaluations. And this works pretty well. It's about comparable to what we're seeing for genomic selection. We actually do really well for solid solubles content, which is bricks. This is good because, everybody likes sugary things, right? And that means that because we have about 80% prediction accuracy in bricks, we're actually going to deploy the NRS as our major phenotyping source for sugar content in the field next year, which is great because just last week I was out there doing the phenotyping with the ground truthing, and the NRS is about 6 times faster than doing it by hand, which means not being out there all day. Our prediction is a lot better in our instrumental traits. You can think about this in a couple of different ways. If you look at flavor chemistry versus just specific sugar content, it's going to be a much more complicated trait. And you can also consider that we're rating things on a 1 to 5 scale instead of 1 to 16 with a decimal point at every single digit. So if you remember, we embarked on this journey just because we realized that, the drone wasn't cutting the mustard for doing flavor evaluation. So, in anticipation of this talk, not really, I had to do this anyways. I went out into the field, I scanned a bunch of fruit, I scanned the leaves in the same plants, and I tried to predict the same trait. And as you see, the novelty of this device is that you just can't really predict traits like sugar off of just the leaves alone. Okay, so now we're going to shift gears a little bit, and we're going to talk about shelf life. It should be no surprise to anybody in this room that's bought a strawberry or had the ones in the room earlier today. They were good, but they weren't fresh. Strawberries don't last very long in post-harvest. So I saw something from the USDA, about 25% of yields from the field get lost before they even get to the consumer. And when they do, they last between 3 to 14 days. We are working within a 10-day window of cold storage and evaluating from there. There's also no widespread evaluation protocol within our program, which might be a surprise to you guys, given how much work we actually do in the state of Florida. Evaluation is kind of just boiled down to the breeder himself taking the most advanced selections home, putting them in a cooler, and evaluating them himself. So we have nothing in our main stage or our early pre-breeding pipeline to actually evaluate this because it takes a long, long time. So I am building out a computer vision model. I'll only talk about one little node of that model today. And we are using that to do image analysis as well as object detection and instance segmentation to hopefully roll that all into a classification approach to approximate how breeders are actually going to conduct their genotype rankings. So I'll talk about the model today's keen object detection, but I also wanted to shout out my very poorly performing bruise segmentation model, which I hope to train a lot better over the summer. This is my first year working with this data set. So we today are just going to talk about gloss. Is it a protective seal or is it just eye candy? I think that gloss would be a great, kind of introduction towards breeding for better shelf life. Humans like shiny things. People like shiny fruit. That's why I brought the watch today, because it's shiny, right? Fruit cuticles also regulate senescence. And, we don't necessarily know. There's conflicting reports on if gloss is going to contribute to this, but it is a part of the fruit. So we hope we can indirectly select in one way or another. We also see that, from this, gloss is heritable and predictable. We see a pretty decent narrow sense heritability just for a single season's worth of data, about 40%. Our genomic selection works fairly well, and our phenomic selection is working even better. So we're excited about that. And just the pipeline is, I go into the field, I collect them into single-celled egg carton trays so that I can phenotype the exact same fruit pre and post-harvest. I enter it into the imaging pipeline through a controlled imaging box, and then I use an NIRS to scan the fruit, and then I evaluate it visually, as well as with a gloss meter that I use to measure the actual gloss. So this is from time zero gloss. So one thing that I noticed when I was working through the data was that some fruit that appeared very shiny got very low gloss scores, which I was not excited about. The whole excitement with the gloss meter is that I personally have a very poor breeder's eye, and that's why I work with the numbers. So I was hoping that the gloss meter would solve all my problems. I wouldn't need to put it on a 1 to 5 scale, but I found out nothing is perfect. Some of these fruit that seem to have a lot of achenes have very poor gloss, and I figured that the achenes were actually going to be occluding what the gloss meter is actually able to capture and what the NR is able to predict. So I trained a just super lightweight, only a couple hundred images, YOLO detection model to detect all of the achenes. I fed that back into our PLS regression, and I improved prediction accuracy by almost 10%. This is pretty exciting. This is a smaller data set, but if you consider the full data set where we're at about 60% prediction accuracy, if we can bump it up to 70 by including computer vision models to help the NIRS models, that means that we'll actually be able to deploy this trait like we are for bricks, which is pretty exciting. So now we're going to get a little Silicon Valley on you. So objectives 1 and 2, there's a lot of questions to be answered. I thought of more even on the drive-in this morning. But in objective 1, if you think about genomic selection, if we're looking at this as phenomic selection as analogous, genomic selection, you can stack your data over multiple years. You can increase your training set. It works very well. With PLS, we're not sure if that'll be the case. We're collecting a second year of data now. Is there going to be a retraining requisite every year from our phenomic work? Additionally, we started working in the end of last year and as well as this year doing metabolite analysis, so GCMS, to get flavor metabolites. And if we can predict those, can they have improving effects on how we're going to predict sensory as well? So objective 3, this is where it gets a little confusing. Shelf life is very complex. You know, you have the visual aspects. Does it meet the USDA grade? Is it riddled with disease? Is it water soaked? Is it bruised? Is it gross? But you also have, what does it taste like? You have to integrate those two things into a trait. And there's a lot of different ways that you can actually slice that up to evaluate that. So what are the latent features that we think contribute to shelf life the most? This can be done, in my opinion, one of two ways. We'll investigate them both. One will be just using a multi-trait index or correlations to overall evaluations of appearance or flavor. Another way is using unsupervised neural networks to try and find those latent traits and evaluate based off of that. Then, in the event that we have a lot of data, which I hope that we will by the time that I'm done with my dissertation work, can we use neural network classifiers to actually approximate how breeders are going to rank things using a traditional multi-trait index? So we will have metabolite data, image data, spectral data, as well as breeder evaluation data. Can we annotate those all into a neural network, assign that a class of 1 to 5, and will it be able to do a comparable job to a breeder, how they would actually evaluate the fruit overall? And then I wanted to acknowledge my committee, my chair, Dr. Vance Whitaker, and everybody else on board, internal collaborators within our group, especially the program staff that, you know, keep the fields going and pick the fruit, help me with the data, all that stuff, and then funding sources as well. And of course, most importantly, I wanted to thank you all for your time today, especially since it's early in the morning. So thank you.
Once again, if anyone has questions, please. I'll start off. So you mentioned that obviously as humans and consumers, we prefer glossier, shinier fruits. Are there major pests in strawberry production that also prefer glossier fruits?
That's a good question. I can't tell you that, but I can tell you we had a terrible year for pests this year with thrips. I'm not sure. I will have to look into that. But I do know in general, if I think I used to work in trees, that if you have a waxier leaf coat, a lot of the times that means that there's going to be terpenes and things that the bugs don't like. So, you know, hopefully as we breed glossier fruit, we might be able to limit some of that. But from what I can tell you this year with our current gene pool, I don't think it matters very much. The bugs were all over us this year.
Great. Fantastic talk. I guess one of the questions I have is you're talking about phenomics, but it seems like a lot of what you're trying to do is actually measure something on the plant. So if you've had drone, do you guys have drone data for
Yes.
these? You know, part of the phenomics methodology would be, you know, you're trying to collect as many features as possible and use those as prediction, like genomic prediction. Have you guys tried to use vegetation extracted features to predict some of these same traits?
Yeah, so we only have worked on bricks for vegetation indices, and it does not work very well. Outside of that, I don't work with the drones directly. We have another lab that does that kind of work. But from what we've seen with that, we've just decided that it's better to try and work with things that we can do on site.
Okay.
Thank you.
Okay, great.